Hi everyone and welcome to our summary on amino acids. So what we're going to do in this little video is go through these couple of spec statements. We're going to make sure you can understand the general structure of the amino acid and also how we're going to form dipeptides and polypeptides and how we break them then down. So first thing, one of the questions they can ask every once in a while is about the composition of these different biological molecules. So when it comes to our amino acids, there are 4 elements that all amino acids contain. So they all contain carbon, hydrogen, oxygen and nitrogen, and there are some that contain sulfur. Not all, only some. So remember, all amino acids, carbon, hydrogen, oxygen and nitrogen, and then there's some that have sulfur in them. When we consider all of the amino acids that are going to be used to make proteins, then it's actually only 20. So we may think with all of these different proteins that make us up as a human being and all these other organisms, there must be huge numbers of amino acids. There's not. There's 20 of them. Now, what we actually find is, even though there are 20 amino acids that all have their own unique little feature, the majority of the amino acid molecule itself is the same. They've got this same basic structure, and you need to know this structure. They could just ask you to name certain parts of it. They could ask you to actually finish off part of a displayed formula. So when it comes to our amino acids, they've got 2 groups. So the first thing they've got is the amino group, which is the NH2 part. And then they've got a carboxyl group, which is the COOH part. So what we have there are the 2 key parts of the amino acid. When it comes to actually drawing this amino acid, the way you're going to do it is you're going to start with a central carbon atom. From there, we are going to have 4 bonds. So we can put our 4 bonds in straight away. Now, onto one of those, just put a hydrogen. Then what we need to do is add our 2 groups, one onto each end. So if we put on our amino group first of all, start with a nitrogen, and onto that we need 2 hydrogens. So there we go, and that is our amino group. On the other side, we're going to start with the carbon atom. We've got an oxygen and a hydrogen, and then we need a double bond going to another oxygen. So we've got COOH, and that is our carboxyl group. Now we've obviously got this empty little bond down here on our carbon atom, and that is going to be R. Now R just basically stands for the rest of the molecule. This is our side chain, okay? Now the side chain is the bit that's going to vary in the different amino acids. The rest of it is going to be the same. So always start with your central carbon atom, then you can add on your amino group, NH2. On the other side, put your carboxyl group, C, double bonded to an oxygen, and then bonded to an oxygen and hydrogen. Add the hydrogen, and then add the R group. So make sure you can draw that so that you get it right if it asks you on an exam. When it comes to the names of our amino acids, the vast majority of our amino acids all have a name that ends in INE. So if we think of some of the amino acids that you've hopefully heard of at this point, things like glycine, cysteine, proline, all of them have that INE ending. There are some that end in the word acid. And the reason that these amino acids end in the word acid is because that R group, that side chain, is acidic. So if they've got that acidic R group, then their name is going to end in the word acid. Why do we care about this R group so much then? Well, the simple answer is that it's that R group that's going to actually give a whole range of properties to the amino acid itself. So what we'll find is that some of the R groups on those 20 amino acids are going to be hydrophobic. If they're hydrophobic, when it comes to folding up into that final protein structure, then they're going to orientate away from obviously any watery environments. If they're hydrophilic, then they're going to turn outwards towards that watery environment. So what we find is, depending on what that R group is made from, that's going to be very important when we come on to see how these amino acids then form that final protein structure. One word that we should know is the phrase amphoteric. Now, when we refer to something being amphoteric, this just means that it's got both acidic and basic properties. So if we break down the word amphoteric, then we've got that first part, amph. So what else have we heard of in words in our general knowledge that have amph? Well, you've probably heard of an amphibian. So things like frogs, for example, live on both land and water. You may have heard of those amphibious vehicles that the army use. They can obviously drive on land and they can also basically go across water. So it's that idea of both. And in the case of our amino acids, the amphoteric means that they have both acidic and basic properties. Why? Well, because as we've already said, we've got that carboxylic acid end, the COOH. Because it's got a carboxylic acid end, it can act as an acid. So just like you hopefully did at GCSE in your chemistry, if we've got our COOH here, that can then dissociate to form a COO- ion and our little hydrogen ions, our protons. Now that means we've just released protons. It can act as a base because on the other side of our molecule we've got that amino group, the NH2, and that one is capable of accepting these hydrogen ions. So we start with our NH2 and it can then accept a hydrogen ion or a proton, and therefore we make this NH3 group. So what we find is the two ends of that amino acid behave in opposite ways. One side can donate hydrogen ions, the other side can accept hydrogen ions. The reason that that is important is because if we've got a chemical that is capable of both accepting and donating hydrogen ions, it can act as this substance called a buffer. Now, buffers basically are things that are able to keep the pH pretty much constant. So if we've got our amino acids present in a solution with a low pH, and remember if it's a low pH that means it's acidic, so there's going to be a greater concentration of hydrogen ions, then the amino acid is going to accept those hydrogen ions onto the amino group to form the NH3. That means we've removed the hydrogen ions from that solution and therefore raised the pH. If we've got our amino acid in a solution where we've got a high pH, so obviously an alkaline solution here, then we've only got a very low concentration of hydrogen ions. Therefore, if our carboxylic acid then releases the hydrogen ion, we are then going to have a greater concentration of hydrogen ions and therefore the pH is going to drop. So what we can see is our amino acids now have this ability to regulate pH and therefore help these living organisms to resist these large pH changes, because they're either just going to donate hydrogen ions or accept them depending on the situation they find themselves in. The next thing we need to know to do with our amino acids is how they join together. We're going to go on to have a look at a couple more videos in this series looking at what actually happens with the proteins themselves. But let's start with the most basic point first of all. As we know from our earlier videos on the other biological molecules, naming bonds is important. So if you are asked about the name of the bond between amino acids, then it is always going to be a peptide bond. So between amino acids is the peptide bond. and that is just a covalent bond with a fancy name, okay? Just like we've seen with all of these other biological molecules, if we want to know how that peptide bond forms, how we join these two amino acids together, then we're going to our old friend, the condensation reaction. If we have a look at our first example then, we're going to have a look to see how these two amino acids join together to form a dipeptide. So, what we're going to do is we're going to look at the example of a dipeptide forming. So at the top you can see that we've got our two amino acids. So this is our first amino acid, here's our second amino acid. So what we can see, we've got our central carbon atom, which has got the hydrogen and the R group attached. We've got an amino group on one side and the carboxyl group on the other. Same in both cases because the only thing that changes, remember, is the R group, and we've just written it as R for simplicity. When these two amino acids join together to form this dipeptide, then we need to be able to draw that peptide bond. So how do we do this? We start off by actually drawing one side of the structure. So all we're going to do is just recreate the diagram at the top, starting from this little bit here, and just come along to include this carbon which has got the R group and the hydrogen attached. Then just stick your little bond out. We're going to do the same thing on the other side, focusing on the other end of amino acid 2. So we put our carbon in place, the R group, the hydrogen, and that joins on to our carboxyl group. Now, we need to know what happens in the middle. So what we're actually going to see, condensation reaction, remember, that means that we're going to be creating a molecule of water. So that means we need two hydrogens and an oxygen to be lost. We can then ascertain the fact that we're going to have this nitrogen still attached. So put the nitrogen in, and that needs to join on to the carbon on the other side, because that's not lost either. What we now need is one oxygen and two hydrogens to be lost. The oxygen and the hydrogen comes from here, and the other hydrogen from there. So that means when we're drawing it, we still have our double bonded oxygen here, and we still have one hydrogen there. That then gives us our dipeptide, linked through our peptide bond. If we go the other way, and we're going to break down our peptide bond, so we're going to break our dipeptide into the individual amino acids, then this is our good old friend, the hydrolysis reaction. And hopefully we remember hydro, water, lysis, splitting. All we're going to do is we're going to add in our water molecule in order to break that peptide bond. And once we've done it, we end up with our two amino acids in that same standard form. So really straightforward in terms of if you know one, you can reverse it to get the other. Do make sure that you remember which reaction is making and which reaction is breaking those peptide bonds. Now, when does this hydrolysis reaction tend to occur? Well, the key area is actually during the process of digestion. So we don't have all of the amino acids we need able to be synthesized in our body, okay? We've got to take some in, and that comes from our food. So that's going to be taken in not just as amino acids. No one just eats amino acids, that's weird. We eat proteins. We then digest those proteins by carrying out hydrolysis reactions on them, breaking the peptide bonds, and therefore we release those amino acids. Now, the enzymes that carry out this digestion of protein, hopefully remember from our GCSE, are proteases. So remember the first bit of the word, what we're breaking down. Ase tells us it's an enzyme. So protease breaks down proteins. We do need to remember that proteins are polymers of the amino acids. Those amino acids are the individual units that make it up, the monomer. So this is again one of those examples of multiple choice questions that come up from time to time, where they will give you all these different biological molecules and ask you to identify the one which is not a polymer. Proteins are polymers, so make sure you know that, and their monomer is the amino acid. The last thing we're going to have a little look at in this video is just a quick overview, and it's a very brief overview today. We're going to go into more detail in our future videos about what we use proteins for in our living organisms. Now, these I'm hoping are going to be familiar to you from either GCSE or from other work you've done in your A-level biology course. First thing, when they fold up, they're going to have this very specific shape. Now that means these are going to be incredibly important molecules to us when we consider things that need a specific shape. They're enzymes, the antibodies, the hormones, all of those are protein molecules. We're also, when we look at our membranes topic, going to see how they form these either carrier proteins or channel proteins to allow other substances to cross our phospholipid bilayers. And finally, we use these proteins to build up structural components like the muscles that allow you to move your body. So that's just a very brief overview of what proteins actually do in living organisms. We will of course look at these in more detail throughout the rest of the biology course. As always, I do hope that you found this video useful, and don't forget that you can get extra resources over on the website, and of course subscribe to our channel so you can see when new videos are uploaded.